We have a debate on aspects of M&D to follow over the next, I think we should take 20 minutes, unless I'm going to get a hook from the back, and we'll finish, aim to finish up at 4:40. If you absolutely need to exit at 4:30, we'll understand. But I think given the scope of the presentations, the level of interest here, and perhaps tuned in from elsewhere, we should allow 20 minutes or so for questions. So I would like to invite questions, comments from the floor. We've got roving microphones, and I'd like to take this gentleman here first of all, and then this gentleman in the second row, and we'll try and keep the pot boiling. I'll come back to you, sir, later on. So thank you. And if you have a question for a specific member of the panel, please identify. Otherwise, we can go at random, so to speak. Okay, thank you.
Hi, Professor Hardiman. I'm interested to hear you say that you had a specific test to see how long we would live for. And you said that you could predict, Professor. Is that being used at diagnosis? And at which stage during the disease can it be employed?
So I think an experienced clinician who sees people with motor neuron disease will have a reasonable sense when we see people once or twice we'll have a reasonable sense about whether this is going to move very fast or move fairly slowly. So what we did was we took 6 or 8 different things that we could measure in the first or second visit, and just did a sort of a mathematical model that provided within there's a margin of error as to how long somebody is likely, whether it's going to be very fast or very slow. I'm not sure that is particularly helpful in a clinical setting to do that, but it's very helpful if we're recruiting people into clinical trials, because we can then make sure that we have the same proportion of people in each arm of the trial, whether somebody is on the treatment arm or the non-treatment, the placebo arm. Because if sometimes we can make a mistake in trials and people, the arms aren't fully balanced, as we might get either an indication that the drug is working, that where it isn't working,
sorry,
or the opposite.
I didn't think it was a drug. I thought it was...
No, I'm saying if we're putting people into the clinical trial. So yes, it is possible to do this outside of a trial setting, yeah.
So there is a test, you're saying
it's not a test.
Questionnaire.
No, it's not a test. It's
a questionnaire that you fill out.
No, it's not. It's not a test and it's not a questionnaire. It's kind of an algorithm. So it's 6 or 8 things, the age of onset, the length of time, but from the first symptom to diagnosis, the rate of progression up to now, the breathing, the 6 or 8 parameters that we can plug into this model. It's kind of a mathematical model, and it'll say, a 50% or 75% probability. So,
and how long has that been available for?
It got published last year in Lancet Neurology.
Okay.
Yeah.
The reason I'm asking is that when I speak to other people with ALS, they're telling me that most of us are told we've got 18 months or 2 years.
Yeah, that's just plain wrong.
Well, it would appear to be, especially in my situation.
Yes.
So can the test be taken, the algorithm be used at any time?
Yes, it can. Yeah.
Okay, where do I sign up?
The reference is there. It's in Lancet Neurology. Hank Jan Vestingung is the first author.
I'm going to remember that.
I'll give it to you later. Yeah.
Okay, thank you for your question. There's a gentleman here, and was there another hand raised in the hall? Yes, this lady here. So we'll take this gentleman, and then I'll come back to you just in a moment. If the gentleman on second row here, you got the microphone working? Yep. Can you help the gentleman there? You ready? Hurry. If not, we'll go to the lady. Oh, I see. I beg your pardon. Perhaps we'll come back to that gentleman.
Miracle study using interleukin-2 in conjunction with riluzole being conducted in France and at the Queen Elizabeth Hospital in Glasgow.
Okay. Who would like to take that from the panel? Pam?
I can speak to that. Sorry, what was the question around the Miracle study? So there is a study of low-dose interleukin-2 happening at various sites in France and the UK, and Glasgow has just recently opened up as a site, which is great. But what was your question about it? Sorry, I may have missed it.
Maybe if we have time at the end.
It wasn't mentioned.
It wasn't mentioned. It wasn't mentioned. That's.
Maybe if we have time at the end, I'll nip back to the back of the room because I have some slides about miracles that I'll be happy to show. If you can spare me 3 minutes.
Yeah. Thank you for raising that. It's a really important development, I think, that Brian had highlighted earlier. So
I think it is really important because for the first time we're nesting into the trial the biomarkers and so on that all has been talking about. So it's a big commitment from patients to take part in that trial because we're measuring what happens in blood and cerebrospinal fluid, and we're measuring what happens on riluzole first for 3 months before the experimental drug. So really important. I think it's simply the time available. We couldn't go into every individual trial, but it is a really important study.
Yes. So thank you for raising that. The lady here?
Hi. Mine's not very technical, but I've been on riluzole for about a year and a half. Not much has happened, but I did notice in your conference there the interesting factor of inflammation, and that brings me to inflammation where people fall, or inflammation on viruses. This stands out in my mind as not being technical, but quite a big factor. And I'm interested to see how you can deal with inflammation. Also shock to deal with people. Has this any effect on how the disease progresses?
Thank you. Who would like to lead off on that question? Pam? Thank you.
I can start, and maybe Ola would add something. So I think it is recognised that inflammatory cells is really important in terms of the progression of MND. It probably doesn't start it off in the first place, but as the motor neurons begin to get damaged, those microglia that we heard about from the stem cell presenters start, whereas initially they may be protective towards motor neurons. As the disease gets going, they can end up being damaging to motor neurons. So the Miracles trial of low-dose interleukin-2 is all about trying to suppress inflammation. And it's been found, interestingly, there was a study from Texas in the United States that showed that people with motor neuron disease who happen to have a high blood cell count of these particular lymphocytes called Treg cells, so people with high Treg cells do better in terms of how the disease progresses over time. And when you increase the T replacement in the nervous system, behave in a more protective rather than a damaging way. And that's exactly what interleukin-2 in the Miracles trial is doing. We're trying to boost the Treg lymphocytes and therefore dampen down the damaging inflammation in the nervous system.
Thank you.
But whether an inflammatory illness actually starts MND off in the first place, I think, is there's no firm scientific evidence, but I've heard lots of patients say it was when I got that chicken pox as an adult that the whole thing started.
Yeah,
Yeah,
I think
and
Orla would
yeah.
like to add,
Come in.
and then we'll come back to you just in a moment. Orla, thank
Yeah,
you.
just to add a little bit on the inflammation, I think that's going to probably be another segregating factor, because not everybody has changes in the inflammation, or there are different types of changes in inflammation we see. So I think that will be one of these factors that we will use in precision medicine. In terms of your question about shock, and emotional shock or injury, there isn't really any strong evidence. Some people think that an injury may be likely to set off motor neuron disease, but the evidence for that, in my opinion, isn't very strong, and I don't think it's really good at the moment. I don't think there's any good evidence to support that at the moment.
Okay, thank you. I think the lady would like to come back. Would you? Do you have another comment to make, just to the microphone for a moment?
Yeah, just a quick one. Speaking to people, we all talk about how things started and how the disease is progressing. I felt it would be a good idea to have a website where people with motor neuron could put their symptoms of how they started, when they started, how they feel. That might be of some use to clinicians in the future. We are throughout the British Isles, and it might just throw something up that might be useful.
So there is a site where you can do that. It's called Patients Like Me, and you can enter into that, and they actually run studies as well. Patients Like Me have run some trials as well using web-based technology, and you can actually measure your own ALS, FRS and put that up. And there's also a chat room on the Patients Like Me as well.
That might be very useful. Thank
Yeah.
you.
So, good question and good suggestion. Thank you. Is the lady to my left in the middle of the row there? Yes, thank you.
Yeah, our experience of trying to get on a drug trial, we've kind of discovered that the criteria are so tight that it excludes so many people, and that's very frustrating. And I'm wondering why can we not lobby pharmaceutical companies to widen it if they're setting the criteria, or can we not fast track patients onto phase 3 clinical trials faster? Can we not do something about this?
Yeah.
Orla, thank you.
Yeah, so I completely agree with you. Part of the problem that we have, and part of the challenge that we've set ourselves in TRICALS, is because the outcome measures that we use at the moment are very specific, like the ALS, FRS and breathing. And they're the measures that are required for regulatory approval at the moment. So they're really, it's unfortunately, it's survival and then how fast you're progressing on the ALS, FRS. So what we need to do is open up, a little bit, think a little bit more outside of the box on this, and think about, like what I was saying about the attributes, that there are many other things that we could measure as long as we have a robust, a proper and valid way of measuring that doesn't have any variability in it. So I think where everybody accepts and agrees that we're far too stringent in how we enroll people in clinical trials. If you look at all the trials that we've done so far, we've tested this in our register, only about 6% of people with motor neuron disease are ever eligible for a clinical trial. So that's not really acceptable. So one of the purposes of TRICALS is to have a trial for everybody, like in cancer. But we have to be better at saying what we want to measure. And that's what I was saying about the biomarkers. We need to have a better suite of biomarkers, better ways of measuring how the drug is responding. So that isn't just about looking at your ALS, FRS or looking at your vital capacity, because people who have bulbar onset disease a lot of people can't do trials at all because they can't do the breathing into the machine that gets you your vital capacity. So we need to move beyond the existing measures that we have into better outcome measures that are more relevant to people, but also that are acceptable, so that we can generate really good evidence that the drug is working, because we have to provide that for the regulatory and reimbursement people But I think that's a challenge we've set ourselves in TriCals, to really invest heavily and invest all of our collective wisdom and expertise and funding into how to make this better and make it more open for people at all stages of their disease, that we can measure something in any aspect of disease. Sort of put up the idea about attributes, that we want to be able to have outcome measures that apply to every single part of the disease.
Okay,
Yeah,
thank
I
you.
think-
I think Brian would perhaps like to
Yeah,
add
I
to that,
just
and then
wanted to
I'll come
add-
back to you. Yeah.
Add a little bit to that, because I'm always banging on about the need for biomarkers. You know, we just don't know what's going on in the inside. That's one of the challenges. And if I can use multiple sclerosis as an example, we've seen huge advances in the treatment of multiple sclerosis, a lot of drug companies coming in, developing effective treatments. And part of that was down to what I would call a biomarker, was the fact you could use an MRI scan to actually see the lesions in the brain. And because you could see them, you could see what was going on the inside, you could effectively do almost like a quick and dirty trial. And if the lesions got smaller, even if it wasn't clear if the drug was working on the outside, you could see it was having some effect on the inside. And that just gives the companies the confidence to then spend the tens of millions that are going to be required to take a drug through the trial process. So, it's one of the reasons that with the Ice Bucket Challenge money, the biggest single project that the MND Association has invested in is actually developing a large biomarker collection from, well, hopefully up to 900 people with MND who will be participating and donating samples at regular points through the course of their disease, so that we can try and identify a blood test, a CSF test, maybe an MRI scan in combination, that will help us to predict how a person's likely to progress. But then if we can recruit people who are likely to progress in the same way in trials, that means smaller trials. Smaller trials mean cheaper trials. Cheaper trials mean more trials.
But I think it's more than that, Brian, as well. I think we need to be able to measure other aspects of what's going on as well. So that, because the constraint, really, the 6% is constrained by the likelihood of having an LSFRS, and that's what we use, the likelihood of that moving at a particular rate. So if we have other ways, not just the LSFRS, but other ways of measuring disease progression that are data-driven, like imaging or this brainwave testing that we can do, they're the corollary of the MRI scan with gadolinium for MS. Although that's not completely fixed yet either, because the secondary progressive phase of MS has the same problem that motor neuron disease does. The MRI doesn't really work there in the way we use it. So the MS people are in the same boat as we are in terms of how do we measure progression, how do we get people with later stage disease into trials where we can measure something to determine whether the drug is actually working or not, because our measure is just not sensitive enough at the moment, and that's the problem. Okay. But I think, just to reassure you, the clinicians who see people with ALS are at one with you on this. We completely agree with you, and we are as frustrated as you, and we want to fix this. And I just to give you an assurance that, those of us who are engaged in trials and those of us who see people with ALS who also engage in science want this fixed as well, and that's what we're going to work on. That's the big challenge for the next 5 years for us.
Okay, thank you. I hope that provides some reassurance for you. Do you wish to comment further? Yeah. Would you like to use the microphone there, please? Thank you.
Yeah, my wife tried to get on the microcals trial, and what the problem was that she was already, when she was diagnosed, you're automatically given Rizil. And that excludes you from the microcals trial. So it took us a little while to realise that, by which time, okay, now she's excluded. So had the person who diagnosed us said, would you like to go on any trials, therefore don't take the Rizil now until such times as you've been rejected from the trial, that would have helped. Okay. Also, there's a question for, if I may sneak one in.
Yes, just a quick question.
Right,
Thank
quick
you.
One. This one's for Dame Pam. We've just been offered a blood test for the C9 off 72 gene to see if there's now, are there any other tests that can be done? Because when she was being tested for that, I said, well, can she also be tested for the SOD1 at the same time? Can she also be tested for any other mutant genes? There's about 30 of them. Why can't you do it all in parallel rather than sequentially? And the categorical answer I got was, no, we do one at a time. Now, bearing in mind that this disease, you might not be around for the second or third test. Is that a sensible way to go about it, or is there a real reason why you don't do it?
Thank you.
There are a small number of centres, and Sheffield is one, that will screen the whole panel of 30 genes. We tend to start with C9 because the change is in a linker sequence and not in the coding sequence of that gene. You test for that in a different way. So we test for C9 and then we do the panel of 30 genes. There's a cost to that, and it costs about, it is, let me work out, because the number of genes has recently gone up. It costs about £1,000 to run the 30 genes. 30 genes? Yeah. That's good value for money, isn't it? Yeah. But I think not all NHS departments accept those costs.
Can I just comment as well, because there's been a lot of discussion around genetic testing and what genes should we test for and who should be tested for what. And there's a very nice piece of work that was done a couple of years ago by the Italians looking at the frequency of the gene variants. And the reality of the situation is that in European populations, the big genes are C9orf, SOD1, TDP43 and FUS. And actually the other genes are of theoretical academic interest. But in terms of what we're going to do, they're actually very, very rare. So they're very useful to model the disease, to allow us to understand what might be happening in terms of the very complicated biology of the human. But in reality, from a clinical practice point of view, those four are the big ones. And in fact, in Ireland, where I come from, we don't have any SOD1 at all. So we don't test for that because it's just not there in Ireland. Our population doesn't have it. So each population is a little bit different as well. So for example, C9orf isn't an important gene in Japan, where there's another gene called optineurin. So the genetic testing also is determined by how the likelihood of finding something, and secondly the likelihood that we might be able to do something with that from a clinical point of view.
But I think there may be further development. So as part of the Ambrosia study that Brian mentioned, where we're systematically collecting samples from volunteer patients, so as part of that, everyone who goes into it that gets the 30 panel of genes screened, and things are coming out quite frequently on that in patients with no family history of MND, but we're finding one of those genes, or sometimes more than one of those genes, is altered in people who appear to have sporadic MND. So I think there are going to be further developments.
Okay, thank you. I think with respect, I'm going to have to move on. Please, could you? Yeah.
Yes.
Don't do it. You're not even looking.
Yes.
Yeah. Well, thanks for your point, and I hope that you can pick up in specific terms if there's any value in that on an individual basis. So thank you for your question. We have questions coming in from further afield. I think we have time. I'm looking to Craig here. I think we have time for one more. And I would like to give that privilege to the audience out with these four walls. And I think Niamh has the question. Thank you, Niamh.
Yes, thanks. I actually have four questions, but I'll just go with the first one that came
please.
in.
Okay.
And this is a question to all the panellists. Lee says, firstly, thank you for choosing to work in the most challenging field of human disease research. How do each of you see the path to delivery of an effective therapy mapping out over the next 5 years?
Just a nice little question to finish with. I think can we give each of the panelists a chance to take a stab at this? And I'm going to give you 60 seconds each. Okay, maximum. Go.
So what we found out in all this meeting is we know that this brain disease is pretty complex. 5 years seems to be a short time, but what we can promise or what we are trying to achieve is with all these tools and developments, we are trying to accelerate whatever we try to do. I mean, there's always an accelerate button than the day before. That's one thing for sure, what we're trying. I don't know whether within the 5 years we'll find something, but again, with the clinical trials or everything, what is evolving, even the clinical trials are evolving. So, and the repurposing of drugs can probably get somewhere.
Okay, Pam, thank you.
So I'm a glass half full person for several reasons. So firstly, I think within the next 5 years there are going to be several gene therapy trials that will come to fruition, and I hope will show positive results for that subgroup of patients. Secondly, I think some of the university academic labs, and we've heard some detail about this from several of the presentations, we can now model to a certain extent motor neuron disease in a dish, and we can screen lots of drugs and potential therapeutic approaches. Thirdly, I think pharmaceutical companies have changed. So they're very interested in motor neuron disease. The market size isn't huge for them, but if we could do for MND what we can do for multiple sclerosis, the market size would hugely increase. And there's great interest now. There's been a sort of resurgence of interest from pharma companies in neurological diseases, and they're linking with academic departments and developing partnerships in a way that didn't really happen before. Mm-hmm. And if you're doing all the bioscience within a pharma company and you don't get a quick result, they kill that program and move on to something else. But the academic labs don't give up, they keep going. And pharma are linking, I think, in a very positive way with the academic laboratories now. So for three reasons, I'm optimistic that we will have made some progress in the next 5 years.
Thank you. Brian?
I'll come at it from a slightly different perspective, working for a research funding agency as well as a patient association. And, you know, in the past, drug companies who were running clinical trials used to kind of think on the patient associations as a bit of an afterthought. They'd start running their big trial and they'd think, oh, maybe we should start talking to some of the patients. Now I find they're coming to us at a much earlier stage. In fact, they're coming to us before they've even got to the early stage clinical trials. And I think that's a great, it's a great sign that they're showing so much interest, but also the fact that it allows us to signpost them in the direction of experts like Professor Hardiman, Professor Shaw, who are absolutely fundamental to key parts of the drug development process. A few years back, we co-funded a lectureship in Pam's Institute, and it was specifically for drug development purposes. And every time I read the annual report from this scientist, it puts a smile on my face because he just has drug companies beating a path to his door. So, you know, I can see, my vision is that we have a conveyor belt. We have a conveyor belt of ideas that are coming out the lab that run down towards being turned into drugs that can be tested in the clinic. And what I've been seeing over the past 10 years is this conveyor belt starting to crank up, and we're starting to deliver a series of things that will get to the clinic. And we haven't been very successful in developing those effective treatments to date, but, you know, some of them are going to work at some point.
Thank you. Arpan, I think the conveyor belt is moving in your direction here.
So I'd like to make a few points. First of all, I think coming to events like this mean that you sort of realise that you mustn't give up, and it's very important for junior researchers like myself. I think we're reaching a critical mass of ideas and findings, and I think one of the exciting things, I think, in the next 5 years will be bringing them all together through collaboration, and we've heard some examples of that here in Europe with the NCALS consortium. And I think I think the next 5 years are about accelerating everyone, putting everyone's views together and coming up with a multi-pronged attack to alleviate this disease.
Thank you. Orla?
So I'm very optimistic. So I think we have a perfect storm in a way. So we have pharma interest for reasons that are around orphan drug and patents, and they've moved from blockbuster drugs into rare diseases, and all of the companies have done that now. And so that's very good for a disease like motor neuron disease, because it's a kind of, from their point of view, it's a prototype. If we can find some ways of slowing down or arresting motor neuron disease, it'll help with other diseases like MS and Parkinson's disease. That's the first thing. So pharma is interested. The second thing is that we're beginning to develop the technologies where we can look at new drugs in a really fast way in the laboratory with the iPS cells. So we can do much cleverer and smarter and cheaper ways of screening new drugs. But I think the biggest thing of all, and the mistake that we made in the 10 years up to about 3 years ago, was that we've recognised what a mess we've made of clinical trials. And I think the next 5 years is going to be fixing that mess. And I think that we'll be much cleverer about the way we design clinical trials. They'll be cheaper and smarter and more inclusive. So in the 5 years in the next 5 years, I see motor neuron disease being divided into constituent subgroups. Not all of them. We won't have fixed all of them. We'll definitely have treatments for some of the genetic forms. We'll have much better outcome measures. We won't be constrained by the ALS-FRS slope and vital capacity and survival. We will have other things that we can measure. The work that Brian is funding, the work that others are doing in biomarkers, both for diagnosis and also to look at disease progression, and also to look at whether the drug is getting to where it should be and whether it's affecting what we think it's affecting. So I think we'll have imaging tools, we'll have neuroelectric brainwave electric signaling tools, we'll have something called PET scanning tools, we'll have a lot more technology. So we'll also be able to get people at different stages of their disease into trials. It won't be just 6%, it'll be 6% of people who can't get into trials, and we'll have 94% of people who'll be able to get into trials. That's what I think. That's my vision, but I think we can make it happen. Thank you.
Thank you very much. That's a really good question in which to wrap up the discussion. And I've just got a few brief closing remarks. I recognise there are hands raised right across the hall, and another 3 questions at least from further afield. So I hope that you'll recognise there are mechanisms for you to pursue these questions through the patient interest groups, perhaps as Brian was suggesting, offline once the formal session finishes. So please take your questions forwards. So thanks for all your interest and participation. Firstly, I'd like to specifically acknowledge Craig Stockton, his team at M&D Scotland, and indeed the International Alliance for coordinating this event. Thanks to everyone in the hall for managing the session, the IT and so on, the external links. Particular thanks to everyone in the audience for your attention, your participation. We were dependent on your reaction, your feedback, your questions to make the session a success, and clearly by that measure it has been a resounding success. So, and lastly, I'd like just to take the opportunity to thank once again our experts for giving their time, their knowledge and their experience for our benefit. I hope there was something for everyone this afternoon. I've certainly gained a huge amount myself, and I hope the same is true for each one of you. So perhaps you could show your appreciation in the usual manner. Thank you very much.